Hi, everybody. This is part two of chapter three. We'll be continuing where we left off on the endocrine system. The endocrine system is a network of glands that produces and secretes hormones into our bloodstream. The system itself is distinct from the nervous system, but it does work closely with, especially related to the limbic system, to help us regulate our thoughts, emotions, and behaviors. Some of these main glands in our endocrine system include our thyroid gland, which regulates bodily functions such as temperature, heart rate, as well as the adrenals, which regulates stress response, our pancreas, which controls digestion, and the pineal gland, which secretes melatonin, for example, influencing our wake-sleep cycles. Remember, the hormones we talk about here are chemical messengers that influence several basic functions like metabolism, growth, and sexual maturation. And the main glands are really the thyroids, the adrenals, pancreas, and pineal. The pituitary gland is referred to as our master gland, particularly of the body's hormone producing system. It releases hormones that directly influence the functions of many other glands in the body. It helps to control stress, digestive activities, and reproductive processes as well. Here you can see, I'll try to zoom in for a minute, where in the brain the hypothalamus is located with the pituitary gland underneath and the pineal gland behind, our thyroid on our neck, the adrenal glands above the kidneys, our pancreas, and then we have our sexual reproductive organs as well. The brain itself is divided in the cerebral cortex into four different lobes. Each hemisphere in these lobes contain gyri and the fissures themselves. So the gyri is referred to as that smooth portion we can see when we look at our brain, and then we can see these sulci or sulci, which create these grooves in between different areas of the folds of the brain. The cortex itself has the highest level of brain function, and it's organized throughout each of these systems themselves. So when we think about what are the four lobes, the first one will start as maybe counterintuitive to how we think it works, but the occipital lobe, what's in the back of your head, which is responsible for vision. So when we see, even though we know we're looking and opening our eyes to see the world, vision begins in the back of our head in the occipital lobe. The temporal lobes on the sides of our heads are responsible for things like memory, the initial processing of things like sound. The parietal lobe tends to deal with premotor and motor activity, and the frontal lobe is responsible for executive control, organized thought. Really, the you of who you are. We'll get more to that when we talk about Phineas Gage shortly. The left and right hemispheres also exhibit contralateral control. That means the left hemisphere controls the right side of our body, and the right hemisphere controls the left side of the body. If it was the same side, it would be ipsilateral control. So these hemispheres, the left and right hemispheres, are connected by commissures. The largest one is called the corpus callosum, which is a thick band of nerve fibers that connects the cerebral cortex to each other, meaning they're sharing information to each hemisphere via this structure. We know this because if you sever the corpus callosum, it effectively creates two brains. This is typically done to treat abnormalities like seizure disorders. But you have to imagine if you have two brains, one, it's not necessarily as noticeable as you think. In fact, people that have had this can still live largely a normal life, but they can also do some things that you can't. If you hold your left and right finger on a table and you try to draw a star with your left hand and a square with your right at the same time, you'll notice you won't be able to do it. You'll try to do it with one finger and the other one's not moving. But people who have had split brains performed or the corpus callosum severed, they can actually do both at the same time without making an error. It's pretty cool, but it's also at a cost of the two hemispheres not really being able to talk to each other. We'll show us some images of um, objects presented to patients like this and some of the challenges they exhibit trying to locate objects in the world. Organization within the hemispheres. The occipital lobe processes visual information, as I mentioned earlier. The parietal lobe processes information about touch, as well as motor control, pre and post motor. Temporal lobe is about responsible for hearing, language, but also for memory. The frontal lobe, I told you, is specialized really for executive control, high function behaviors, movements, abstract planning, um, memory, and judgment-based decisions. The somatosensory cortex is really between, we talk about the parietal and the frontal, represents the contralateral control of our bodies. So the left and right hemispheres controlling the opposite right and left sides of the bodies. When you looked at these particular areas, what you'll find is that while there are areas in the motor cortex that are responsible for regulating movements to our hands and our legs, for instance, if you look over at the homunculus map, which is effectively mapping the human body to these areas, you'll notice there's a great big face staring at you on the right-hand side with its mouth wide open. Now, the proportion of the body parts that you see, meaning why some are larger than others, represents the number of receptors that our brain receives inputs from the body. Meaning we get lots of receptors from information from our face, like our cheeks, but our lips in particular, as well as our hands that send a number of information to be processed to the respective hemispheres in our brain. We also see smaller mapping size of our arm or back or our bodies, the main trunk of our core. Because even though there are receptors there, there are fewer of them compared to like our fingertips, as an example. So this here map really does somewhat accurately capture the differences of inputs that our brain receives from the senses in particular. We also have association areas. These are composed of neurons that help provide a sense and meaning to information registered in the cortex. It really helps stitch together information, especially in the cortex. These threads can produce meaningful understanding of what is being registered in the brain. Let's take, for example, the mirror neuron system. In the early 90s, I think it was 1992, the Italian colleagues were trying to investigate if neurons in the monkeys, it's called area F5 of the macaque monkey. It's similar to your Broca's area, which is left frontal lobe. If the area F5 would respond when the monkey reaches out and picks up a peanut off a tray, which they were able to document that it does. So you can see that there are certain cells responsible for a monkey reaching out to grab some food off a tray. But they also noticed that the same cells fired when the researchers reached out and touched the food. So as they ate it, you'd have to replace the food. And the same area of F5 of the macaque monkey was firing when the researcher reached out and grabbed it when it wasn't moving. So when it watched behavior, the neurons also fired similarly to when they actually performed the behavior of reaching out and grabbing it. The idea that neurons would mirror activity in the real world was quite fascinating. Think about what would be the purpose of maybe neurons firing in the absence of movement. Maybe we see somebody um, who hurts themselves, and we might feel empathetic towards the pain they're feeling. Maybe parts of us kind of feel that minor ache or ailment in watching others suffer. Is that related to mirror neuron activity? Possibly. It's been one of the arguments made why kids in daycare start crying and then everybody else starts crying with them. To be empathetic to the other children who are actually hurt. The majority of the work really focused on macaque monkey. Now, it's argued that humans have mirror neurons in what we call Broca's area, the extent of which is not as widespread as you might think they are, considering the importance. But nevertheless, largely we think about it as when an animal observes another animal engaging in a specific behavior, some of the neurons in the observer are active similarly to the ones exhibiting the action. And there's been lots of social arguments made for why this would be advantageous. There's much more I could say about it, but let's just leave it simply as that. What's really important to think about as well, and I get we can't really have that explicit dialogue around plasticity, but it's an odd concept to think of your brain as plastic. Really, you need to think of it as it's malleable. It is subject to change. Like, let's take right now. Think about your brain. Think about the information you know. Is your brain today the same as it was yesterday? Will it be the same as it is tomorrow? And the answer is no. The connections that you form through learning are indicative of a brain that is constantly changing. If you know something new today or experiences that you've had that are, you know, forged in neuronal firing in your brain, then this effectively is what we call plasticity, your brain's ability to change. And that change is in dendritic growth. So when new neurons form connections, you just get dendritic growth. Our sensory cortices are not fixed. They can adapt to change. So it's really the effect of that our genes produce the ability for these neurons to grow new connections, as well as the environment to provide us the opportunities to change them. Different functions can be assigned to different areas of our brain, and they may be capable of being reassigned to others in the extent that there's damage that can occur, or really a change in input. The best example I can think of this is phantom limb. In this instance, I believe it's somewhere around 78% of individuals who have suffered some form of amputation result the experience of phantom discomfort, phantom pain. And as a consequence, you can see how the brain, the cortex, can remap itself. So if you're missing an arm, for example, it's not uncommon for some people to describe that their hand that they're missing is really painful. It's hurting. It's like their fingers are all twisted, or they have something like an itch. Some ways to alleviate that itch is that with your where your hand is mapped in your cortex is similarly to your cheek. And for some people, it's like you can scratch their cheek and that itch goes away. There's also mirror-based therapy you can use with phantom limb pain. You can just effectively put the mirror in front of a person and have them put their left hand out, but look in the mirror and see your right hand, and you can stretch your hands, or you could get someone to help scratch. As long as you can see it, it just tricks your brain into believing that the ache is now going away or the itch has been satisfied. But ultimately, it's suggesting that our brains are capable of reforming maps. Now, earlier, you're going to say, well, you said that the peripheral nerves don't regenerate. No, we're not talking about different areas regrowing or returning. No, we're talking about adjacent areas assuming some of the duties that another area did. That is typically true when damage occurs in really young animals or people. As we get older and our experiences really shape who we are and who we become, brain damage can be rather challenging, bordering catastrophic, depending on what the nature of the damage is. So the size of it, the location of it. Ultimately, other areas can't just assume the role in its entirety of another area. But if you're young enough, your brain's still wiring. It's amazing what it can overcome. There's one young boy who doesn't have a left hemisphere at all. The cerebral cortex has been removed, and he can still speak. He can still do other things and has a just below normal level of intelligence. It's really remarkable in a sense of how plastic our brains are and what we can accommodate. Um, Also, the changes that can occur in the absence of things as well. So there's lots of remarkable features about what our brains can achieve and what they can do. And even in the case of phantom pain, it's almost kind of silly how you know that something isn't there, but you experience something in the absence of the thing, your arm, um, is baffling, but still fascinating. I did talk about phantom limb. I mean, I guess I can just mention the evidence of the other areas becoming active are in the fact that we can use imaging techniques to assess their brains, and see different areas lighting up that typically were responsible for receiving inputs from what would have been your hand now into your face. Mirrors are really effective at helping individuals overcome the unfortunate experiences they have of pain in a limb that doesn't exist anymore. So you can see here how a cotton swab to the face can help alleviate some of the discomfort someone would feel in a hand. This gives you an idea of what could happen. This does not guarantee that it would, but it can happen, which is a result of our brains being plastic and remapping. Think about practice. I mentioned in the previous lecture that if we learn a new skill, over time our brain is becoming more, let's say, efficient and effective at eliciting motor commands to allow us to play an instrument, for example. But what about driving a car? Taxi cab drivers in London, there's evidence that, especially in more senior cab drivers, more experienced cab drivers, that they have a significantly larger hippocampus, which is important for spatial memory, if you recall, than novice taxi cab drivers, early career taxi cab drivers, or just the general public. Now, this was true of London cab drivers, but there's a reason for that. In London, they have to go to like a college where they learn to become taxi cab drivers. They study the city, the areas of interest, if you're going to drop off people, tourists especially, and then what else is in the area so they can recommend places to go. They're not supposed to take you for these long-winded drives around the city to get to your destination. They train because they're supposed to get you there and back as quickly as possible. So the most direct route. So they have to know how to navigate the city. Learning how to navigate it has resulted in a change in the hippocampus, which is not surprising because we also see this in food storing birds. Some birds that live in more harsh climate conditions also have a larger hippocampus than similar species that live further south or closer to the border in the northern hemisphere. There's less reliance on having to find food when you live near an abundance of it than living in the absence of it. Exercise can also be really important for our brains for a whole host of reasons, but we'll keep this stuff simple. Basically, can help increase the number of synapses that are formed and promote the development of new ones. This is true in the hippocampus, which is also critically important for memory and our motor functions itself. It's really brain-derived neurotrophic factor they're talking about, but they just don't mention it. But what about culture? Findings from cultural psychology and neuroscience studies have provided growing evidence that culture does exert a notable impact on our development, which is not surprising. Alzheimer's disease, so we really switch from parts of our brain that are responsible, we talk about, for plasticity and some of the experiences that can occur as a result of it. But what about something on the opposite end of it, like forgetting, where now our brain is not functioning the way that it should or it used to? Personally, my grandmother had dementia, and it's not uncommon, as Alzheimer's is the major form of dementia. The challenge when it comes to things like this is that there's no test that can detect it. It's a cognitive impairment that occurs largely due to plaque buildup in your brain. It's not confirmed, and postmortem results indicate that the plaques are present. Alzheimer's disease is more prevalent among women than men, especially in many regions of the world. Why do you think Alzheimer's then is more newly documented throughout history? Why do more females have it than men? You could probably guess the reason to the last one. The fact is, most women live longer than men, so it's probably more likely due to just the fact that women live a longer old age life. The other reason we don't typically see this historically is because we didn't live as old as we do today. So modern medicine, better environments, food, it's helping us to live to ages we historically have never lived. And as a consequence, we can see that our brain, not everybody develops some form of dementia, but um, when they do, it can be pretty devastating, including forgetting not who you are, but forgetting significant things about your life. Your kids, you can be forgetting. Basically, the rule is last in, first out. So all of the recent memories you have start to fade away. And it just seems to erode decade by decade by decade. Um, Unfortunately, until you forget a significant part of who you are and the important things that really were to you in your life. When we think of genetics, you've all had opportunities to learn about this in high school. So, what are genes? Our nature is our biology, and our nurture is our environment. And we're born with, a genetic, you could say, predisposition for some behaviors and things. But really, it's the environment that can help elicit the not just a likelihood of genes being expressed, but think of reinforced neurological pathways for various behaviors we have. So a gene itself is just their unit of hereditary transmission, sections on strands of DNA organized into chromosomes. And your chromosomes are just strands of DNA that are wound around each other in a double helix configuration. We have different degrees of relatedness. Typically, in research, we'll study monozygotic versus dizygotic twins, so identical versus fraternal twins. And then heritability is a measure of the variability of behavioral traits among individuals that can be accounted for by genetic factors. So if we think about, twin studies, I've just jumped ahead for a second, but I'll go back so I don't forget. Think about the information here, degrees of relatedness. Twin research can be critical for a whole host of reasons. Some of which are that, unfortunately, in the past, twins have been separated at birth and raised apart from each other, not just from their parents. And in those instances, there are striking similarities that researchers have observed between the twins when they're reunited many, many years later. It's not surprising that they're much more alike than different, given that they're 100% genetically the same, versus fraternal twins are just related similarly to any other sibling that you may have. 50% of your DNA is the same. The neat thing is that, and I mean, it's neat in an unfortunate instance of twins raised apart, is that the only difference is the environment. Even when you raise twins in the same household, genetically they're the same or they're related like siblings, but the environment's the constant. It's the same for them. So it does offer sometimes unique opportunities to study what is the effect of the environment? What's the effect of genetics? We know things like intelligence are highly related to your genes. You'll be more like your biological parents' intelligence than you are if you were adopted. Since many people have adopted individuals, studies have consistently demonstrated that their intelligence is much similar to their biological parents than they are to the adopted parents. And then you can think about other genetic related factors, whether it's something like we mentioned dementia. What's the likelihood you could, inherit that? And the likelihood is not as strong as you think, especially for things like schizophrenia. But what if you're homosexual? Now, we would usually discuss these things in class, but I believe the concordance rate is somewhere like 78%, meaning that 78% of twins that are, at least um, identical twins, if one twin is a homosexual, so is the other. There's a strong likelihood linking the genetics to it. It's not a choice, just to make that crystal clear. But when you look at other factors about genetic inheritability or heritability, sorry, if you've inherited traits, it doesn't always guarantee things would happen, like schizophrenia as an example. But it's also important to look at how the evidence suggests other things occur so that we better understand the nature of behavior, just simply at that. So, epigenetics is the study of the environmental influences that can determine whether genes are expressed. So, just because you have a parent that has schizophrenia, for example, it doesn't guarantee you will develop it. Yes, there's a likelihood it could happen, but it's not just as simple as having the genes for it. Typically, we have to think about what about the environmental conditions that are at play that may warrant the expression then of the genes to elicit something like what we would call schizophrenia. So we have different epigenetic marks, which are chemical modifications to DNA to turn genes on or off. There's DNA methylation, which is adding a methyl group, special enzymes to DNA, and then histone modification, which adds chemical modifications to proteins called the histones that help in packaging DNA. And here you can just see the images that show each of these. It's a nice visual. Just take your time, pause it here and look at it, or look at the slides on your own convenience. So identical twins, which are on the left, again, share 100% of their genes, whereas dizygotic twins or fraternal twins who are on the right just share 50% of their genes. Studies that have used twins for research really do help to understand the contributions of genes and environment on their behavior. What about genes and early life experiences? Now, I was told when I was in grad school that Michael Meany should win a Nobel Prize, and I don't think he's won it yet, but I could see the case being made for him too. Some of the significant work Michelmini did was he found that certain rats like to lick and groom their offspring. And what they found was when those rats that the pups that grow up that were licked and groomed, they respond to stress significantly better, meaning there's decreased levels of cortisol than rats who had pups that don't lick and groom their offspring. So that's what the LG refers to, high and low LG, licking and grooming rat mothers. So early in life, if you have these rats that lick and groom, and for lack of a better phrase, are taking care of their offspring, they don't experience stress the same way as rats who are stressed and their mother doesn't lick and groom them or didn't lick and groom them. So these early experiences are not just restricted to single genes, but they occur more broadly. Really, Michael Meany is making the argument about if rats, lick and groom their offspring, and the stress response is different. Is this argument then similar to some biological pathway where neighborhoods that are created that are unfortunately disadvantaging some children, do they end up themselves becoming disadvantaged? Do they grow up and then have children? Effectively, what I'm trying to say carefully is the cycle of poverty is difficult to escape. So if you're born poor, you're at a disadvantage, you don't have access to the resources that other, advantaged children do have. And as a consequence, you're not able to learn necessarily the skills to escape poverty. If you have children, the cycle continues. Now, I'm not saying if you are born poor that you're guaranteed to stay poor and to be uneducated. Not at all. But I'm saying the odds are stacked against you, that the data does strongly support that if you are disadvantaged, it's very difficult to break that. Not impossible, just it's challenging. So it's important that we consider these lenses through the development of the genes, the expression of the genes in the environments of which we're in. I remember when I when you think about how environments can shape behavior, which you think about shaping your genes, if you will, and having offspring and thinking about their, not that we really think about genes, we think about the environment. Um, I remember asking members of the indigenous community here, because I'm a member of Halapu Mi'kmaq First Nation band, and in conversations with them, discussing the impacts of things like the residential school. I remember one particular conversation, which was that an individual communicated to me their parents didn't know what love was. And if they don't know what love is, how could they give it to me? So to their child. So if you're not loved and then you have children, how do you know how to love them? You don't. So then they grow up never having experienced love the way that maybe you have, and then they have children. This is that intergenerational trauma that can be passed down, similarly to the licking and grooming in rats. I'm not saying that the animal models have to be perfect, but you can see the connection. You can, I think, appreciate the challenges that when people make arguments, oh, the residential schools, they close, they're over, move on. It's easier said than done. Plus, the most recent ones only closed in the 1980s. It's complex, and it's really complicated to think about how to change it. But those are kinds of the big picture problems that we face, and we have to think about it from multiple perspectives, recognizing that the change is just complicated. Behavioral geneticists use calculations based on relatedness to compute the heritability of different behaviors. So, measures of the variability of behavior traits among individuals that can be accounted for by genetic factors. But you have to remember heritability is an abstract concept. It is a population concept. It doesn't mean you specifically, it's more about the larger picture of everybody we're talking about. It is very much dependent on the environment. And just because your parent has something like schizophrenia doesn't mean that you will. It's not a this sort of um, death death sentence. Like, no, it's not fated to happen. It might, but it might not. One of the I'm going I'll caveat what I'm going to say with one of the best ways to learn about brains is through brain damaged people. But I get that. I don't want to sound like you're excited from people who have brain damage to learn from them. No, the the reality is we've learned a great deal about how brains work because people have suffered brain damage. Um, in the in-person section of this course, I've told students that my own father has suffered significant brain damage as a result of a traumatic accident where he fell down stairs and landed on his head. The reality is your brain is you. Your neurons, the way that they work, it all makes you you. And when they don't function right, they alter the reality of well, your or their experiences. Like I will never forget walking in to visit my dad after his accident, and I said to him, hi, how are you? And he's like, fine. So I'm like, okay. He's answering just generic questions, okay. And I'm like, what are you up to today? He's like, I'm going to the OR. I was like, oh, you're going to the OR? What's going on? And he looks at me dead straight in the eye and says, I'm having twins. And I thought, interesting. So I just said to him, what are their names? And he was really confused. I mean, the reality is he was confused to begin with. Like, he's not having twins. The idea he thinks he's having twins, I thought was fascinating because it's so far from reality. It took a long while for his reality to change. Now, he's not even remotely like himself today, but at that time, he was, I mean, really, he just simply had no memory. He didn't know what was going on. And over time, things progressed to be a little bit better, but it's never quite the same. Just we'll leave it at that. So a damaged brain is is different than your normal typical brain. Um, so we really talk about normal as neurotypical, and a brain damaged brain is atypical. That's not normal. A lot of research that we've understood of how brains work in neuroscience has come from brain damage. Two great examples are Broca's and Wernicke's area, as well as Phineas Gage. Broca's aphasia, which this doesn't really talk to, is the inability to produce language. Meaning, if you have a conversation with someone who has Broca's aphasia, they can understand what you're saying. They just can't talk. Wernicke's aphasia, on the other hand, they can't understand what you're saying. They have a comprehension problem. They don't get what you're saying, but they can talk. The talk is gibberish, though. It doesn't make sense, and the words and the sentences that are formed are incongruent. So it's rather unfortunate when you think about two areas that are very close to each other, in the temporal lobe and the left frontal lobe. So Broca's area, I told you, it's left frontal. It's kind of on top of the temporal lobe, whereas Wernicke's is just back a couple inches, um, but in the temporal lobe, not frontal. Broca's area is where we thought mirror neurons are in humans, I mentioned earlier. So Broca's aphasia is the inability to speak, but you can understand speech, whereas Wernicke's area, you can't comprehend the speech, even though you can speak it. In terms of specific damage, Phineas Gage is really not a good example here, but there's better ones I can think of. One is the fusiform gyrus. It's another way to call that is the fusiform face area. Damage to this, the fusiform gyrus, this area, results in face blindness, meaning you'll never see a face again. You can see hair, though. You can see their clothing. Everything in the world's fine. You just can't see faces. Phineas Gage's damage is much more catastrophic in nature, but it was largely to his left eye and then frontal lobe. Unfortunately, what he did was he used what's holding in his hands, a tamping iron. He was packing down areas when they're building the railroad that contained TNT. As he hit the areas packing down the TNT, unfortunately, it sparked off a rock, which, sparks and dynamite, not a good combination. Unfortunately, it made that object, the tamping iron, a projectile, like a missile, and it went up under his left cheek, exited through the top of his left frontal lobe, and launched like 40 meters away. The significance of Phineas Gage is that this is really the first incident of traumatic brain damage where someone lived. And the main takeaway of Phineas Gage's accident is that his friends noted he looks like Phineas Gage, but he doesn't act like him. That's sort of the challenge with brain damage, is that the people that you know and love from that perspective, this makes it really challenging because sometimes they're just different, and I mean very different. Sometimes they're not. In the case of something like the fusiform gyrus, it's just you can't see faces. Nothing else is wrong. But you can imagine how hard it is to identify people in public. I'm not saying it's easy, doable, but without seeing a face, um, people can't even identify themselves when they're shown images of people with the similar hairstyles. They really don't know who anyone is. So you just have to rely on other features to be able to do so. Early work understanding how our brains function um, won the Nobel Prize for lateral processing. So Roger Sperry earned the Nobel Prize for demonstrating the left hemisphere has specific functions as it pertains to verbal language processing, and the right hemisphere appears to be more spatial. Today, we have a better understanding of how the brain processes things like language, and yes, there is still a left hemisphere significance, we'll say, for language production, but it's also distributed through the cortex, left and right hemispheres as well. So it's not just the left hemisphere's language only. I mentioned split brain um, earlier when he severed the corpus callosum. In this instance, you can see where if you stare at a fixation point, so imagine you're this person staring at that black dot on the screen in front of them. If you're staring at that, you create left and right visual fields on each eye. And as a consequence, what you get is that unless they can see everything on the table, in the periphery, not all the information would be transmitted to the appropriate hemisphere, meaning because they don't communicate, they wouldn't be able to identify, like in this instance, what was presented to the left visual field, a key. They're saying a ring because they can't see it. So again, this is really more of that artificial instance where we can demonstrate that there are challenges to having your corpus callosum severed or two brains, split brain patients. They'll fail with chimeric face experiments as well, which you can see here. So it's really showing the importance of having the ability for the left and right hemisphere to communicate with each other. And the reasons that this is ever severed is strictly due to medical need. The chapter is going to conclude with how do we study a brain today? Case studies are instrumental in understanding the normal and abnormal function or typical and atypical function of brains. But it's also really important to know what happens in a normal, typical, or developing brain than it is specifically to think about it from an atypical lens. We can use techniques like electrical encephalogram today, EEG, to record electrical activity in your brain. Because remember, there's the electrical chemical reaction. So we can measure the electricity that's generated in the cerebral cortex using this device. In doing so, we can identify when we detect things like errors. Imagine you heard the sentence, the pizza is too hot to kick. About 400 milliseconds after you heard the word kick, there was an increased amplitude. It's in mostly left, but also right in your left hemisphere on the top of your head. This is indicating that our brain didn't expect to hear the word kick because you probably expected to hear the word eat, but you didn't. These kinds of activities allow us to not just understand how our brain works, but how it processes things like speech and in where amongst the cortex does normal and abnormal processing occur for things like we expect to hear and see. There's other methods that we can use to studying brains. Really, the most commonly used one, if you go to the hospital, because you hit your head, is the CT scan, the computerized tomography. And this can show different brain densities. It's really good at finding things like lesions or tumors. MRI is also commonly used, or functional magnetic resonance imaging. MRI and fMRI are a little bit different and not exactly the same, even though they use the same device, sort of. They can provide a really clear picture of where in the brain things are happening. So this can help localize brain damage or understanding where in the brain or things are occurring. So it gives us really, really great spatial resolution, so the where things are, but really poor temporal resolution or when they happened. PTI is newer in its ability to help show the brain, but it shows amazing connections of the white matter pathways. They're quite remarkable, and there's really more exciting things to come from that particular measure. This just shows you the difference between CT left and MRI right of what brains look like. CT are maybe a quick and dirty way of seeing what's going on, whereas MRI are much slower and significantly more detailed in the images that are generated. So, is a bigger brain a smarter brain? What do you think? Early studies found a positive correlation between brain volume and intelligence. There's indirect and inadequate methods, though, to suggest that this was even true, meaning it's not. It's not really a thing. And what do you mean by bigger brain? It's not so much about the volume as it is about the connections. Remember, we talked about how neurons fire and form connections. We can't lose sight of that. Now, current research does reveal a positive correlation, but before I finish it, correlation is not causation. between brain volume and other measures of intelligence. So modern imaging techniques can show there are some distinctions between it, but again, it's more the connections, maybe the degree of plasticity ultimately that's important than just size. Um, and again, the correlation used here was really poor, even though it's significant, that's not a strong correlation. Functional brain imaging shows activity while someone engages in a cognitive or motor task. PET positron emission tomography is great, but you need to use a radioactive isotope to reveal where in the brain oxygen's being consumed. It's not typically used in research for more for medical purposes. fMRI is the functional magnetic resonance imaging, which is really good at looking how um, the deoxygenated blood travels through the brain. So really, it's a bold response, blood oxygen level dependent response. This is really more of a physics answer, but just think of it as in your brain, we have ions and they carry a charge. And this is a really strong magnet. It's about 30,000 times stronger than the Earth's magnetic field. So, as a consequence, we're able to change how these ions are rotating. Basically, they're all spinning, and the magnet's going to align them the same way. When you turn the magnet off, that's when you do a task. You turn the magnet off, you whack them with a radio pulse, and they start to wobble. You're trying to measure which ones wobble back to their original state the quickest. And you can usually see this because oxygen and how it flows through our brain, the arteries in our brain, will deviate. It's meaning more blood will flow to areas that are engaged or active than those that aren't. So this gives us a really great picture of our brain, of what's going on where. So it can help localize where in the brain things are occurring. They're very accurate. They're just a little bit slow. fMRI doesn't require any exposure to radioactive substances, so that's why we're not going to use PET. It can localize changes in brain activity across briefer periods in PET. It is more used for quickly occurring psychological phenomenon, but it's not, again, usually used for research, I said. Nor is fMRI, just so you know. It's super expensive. The device alone is millions of dollars. You have to house it in nuclear medicine, staff to run it. It is a giant magnet, so you have to be careful when you go in there. And um, yes, it's advantageous. It can help look at some of the relationship between brain regions and how things change over time, which is super useful, but it's a very costly device to use to conduct research. But these are some of the images that we can gather from PET as well as from fMRI. I've had fMRI or I've participated in studies, about 5 of them, and they are quite an experience. And they do give you really, really neat detailed images of your brain. One of the last techniques is newer, and of the other ones we talked about, like EEG and PET and MRI, they're all correlational techniques, meaning we're using them to measure the brain activity of some activity we are controlling. TMS, on the other hand, can actually create virtual lesions, meaning it can cause a change of behavior. For instance, if we zapped Broca's area, so we use TMS and we can trigger a pulse over Broca's area, it can inhibit your neurons temporarily for firing, meaning you won't be able to speak. So, this method can actually mimic brain damage by temporarily deactivating neurons in your cerebral cortex. It can be used in combination with other techniques, meaning we can use this and then put you in fMRI. Ultimately can help provide causal explanations of behavior. So these kinds of advantages are really new and useful in learning more about how and where in our cortex behavior occurs. That's the end of the second part of chapter 3, and also the conclusion of chapter 3. I hope you've enjoyed this video, and we will have more content for you soon. Take care, everybody.